It is so good to see everybody. I think we're going to get started here in just a moment because we have plenty to do here. Yeah, I think let's go ahead and we are going to get Lab at Home started. So first, as always, our learning process is all about respect, respect for ourselves, for each other, and our world. We ask you all to show respect by using kind language and appropriate words in the chat box, as this is a program for everyone. My friend Max is moderating the chat and can disable the chat function for individual users if that is needed. I don't think it will be. All of that said, it's okay to disagree and it's okay to share your feelings. We welcome all ideas that are founded in respect and invite all of you to do the same. So when we have the lab on site, we encourage our learners to lead with their questions and ideas. So I'll be asking all of you plenty of questions and hope that you will share your ideas and your own questions as much as possible in the chat. Today, I want to issue the Bug Out Creative Challenge because we are in the midst of our summer Bug Out celebration. Then Max has some iNaturalist updates for us, and then we will get started on our experiment. So, we are celebrating our summer Bug Out all throughout June. It is our first ever virtual event dedicated to exploring the world of bugs and getting to know our invertebrate neighbors. All through June, we will have special bug-centric labs at home, feature 1-minute bug hunts with Dr. Eleanor on our YouTube channel. And because insect appreciation is an art as well as a science, we will have weekly Bug Out creative challenges with fabulous prizes. So this week's Bug Out challenge is to draw a close-up, head-on portrait of a bug you found this week, or your favorite bug in general. Right. So a lot of times we're looking at bugs kind of at the top of their bodies because we're looking back down at them. But what do they look like if we looked at them head on up close, right? I would love to see a portrait in the medium of your choice, an illustration like that. That would be so, so cool. So we'll have a post on our Facebook page about that tomorrow where you can put your entry. I hope to see those. So the other cool thing we are doing for Bug Out is this iNaturalist Bio Blitz that Max is going to tell us about. I'm going to let him go ahead and share some highlights with us and share his screen.
Thanks, Peregrine. I'm excited to see what hands-on stuff we are doing today. But really briefly, before we get started with all of that, just a quick reminder that we are also looking for the stuff that you have been finding here on our iNaturalist page, on our Lab at Home Summer Bug Out page on iNaturalist. The link to that is all the way up here at the top. It's inaturalist.org/project/summerbugout. I'm sure that Peregrine or I will toss that into the chat here in just a second. And when you get to our page, you can join all the way over here on the right so that you can join our project and get updates about it. Now, a lot of folks have been submitting a lot of stuff to it. You can see we just started last week, and we are almost to 150 different species just of different invertebrates that y'all have found, which means stuff like our bugs, which includes our true bugs, but also spiders and worms and myriapods, which include things like millipedes and centipedes. I wanted to take a second to enjoy, and for all of us to enjoy, some of the incredible things that people like you have found just in this past week, since last Wednesday, here in Durham. So everything that we're looking at right now on the screen are different bugs that you all have submitted to our project. These are all things that live in your city. So we have this margined leatherwing beetle, which is the most observed bug so far, which I think is pretty interesting. A lot of folks also showed us this yellow and black flat millipede here, which I'm going to zoom in on a little bit because this is a really fascinating one. iNaturalist is really powerful not only because you get to help conduct the research by submitting your own observations, but it also helps us in that using this graph that you can see here, you can also see when you might expect to find them where you live. So here in Durham, this graph shows us what months we might see this particular one. And it also shows you further down where in the United States you might expect to find them. So you can see we're really in a pretty small little area here where there are a whole lot of them, even if folks are submitting observations from elsewhere also. So there's a whole bunch, like I said, almost 150 different species of invertebrates that y'all have found just in 1 week here in Durham, and I really can't wait to see what else y'all find and post and come up with, because again, it's just really, really, really cool, all of the stuff that you've been able to find so far. And with that, I'm going to stop sharing my screen and turn it back over to my friend Peregrine.
Wonderful, Max. Thank you so much for that update. It's a really cool platform, and I like it because I get to see kind of what all of you guys are up to in between labs at home. I can kind of see what sort of cool insects you guys are catching here during our summer bug out. So our bug-themed lab at home today has to do with observing bug behavior. So I have a certain bug with me here today, and it's not really an insect. I'm going to have you guys take a look at one and see if you can tell me what you think this is. You guys see him there? What about this guy? What do you think? Do we know the name of this one? Ooh, we have one person saying a pill bug. A pill bug, yeah. A roly-poly. It's so interesting. We call them so many different things. We call them roly-polies or rollies-poly, as Max might say. We might call them woodlouses. We might call them pill bugs. There's potato bugs is another one I've heard. They have a lot of names, and they're pretty cool little bugs. I'm gonna get up close one more time just to let you see them. And I've also got another camera we're going to use a little bit later to take a closer look at these guys. And they're not actually insects. They're a type of crustacean. They're an isopod. So they're more related to crabs than they are to something like a beetle or a grasshopper. So it's really cool to see these guys crawling around on land. And they do actually even still breathe out of gills, so they need nice humid air. I don't know if any of you guys were able to catch any of these guys for our experiment, which we'll talk about in just a second. But we're going to be looking at some of their behaviors. And we're looking at a certain kind of behavior. Well, first of all, wait a minute. What's behavior? What does that mean? What do you think, you guys? What is a behavior? It's kind of, yeah, the way something acts, how it acts, yes. How they react, what you do, yeah, exactly. A behavior is something that you do. And we're looking at a type of behavior called a taxis. And I'm gonna put that in the chat really quickly here, a taxis. A taxis is a certain kind of behavior that has to do with movement. It's a type of movement toward or away from a certain stimulus. Can you guys tell me what a stimulus is? What is stimulus? Have we heard that word before? What do you think? So maybe that's a new one. It's definitely a useful term, right? So a stimulus or a stimuli are changes in the environment, right? So as a living thing, right, living things react to stimulus. They change in response to changes in their environment. So there are different kinds of environmental changes, and there are different kinds of taxises or taxis that a pill bug might perform in reaction. So a taxis is not like a regular movement, right? Like when we think about humans moving around, we don't usually say that they're performing a taxis. Since we have these big brains, right, we usually think of ourselves as making decisions. Right now, it's a beautiful day outside. If I were to go out toward the light and toward this beautiful day, I wouldn't call that a taxis. I would say I made the conscious decision to go outside because I think it would be fun. A pill bug, right, and many other types of smaller organisms or living things, we would say just kind of have a reflex. They do something without really thinking about it, and that's what a taxis is. I'll give you just kind of one example of a taxis in humans. We observe in baby humans and infants. It's called a grasping reflex. And that's something where anytime if you touch the palm or kind of stimulate the palm of a baby, they will instinctively or reflexively close their fingers because it's a pretty good reflex to have if you're a baby. It's a good idea to be able to hold on to stuff. I don't know if any of you guys have little siblings at home and if that's something that you have observed. So the difference between a taxis and an instinct is a great question. A taxis is a type of instinct. Taxis specifically refers to moving toward or away from the stimulus, the change. And we're going to talk about studying two different taxis in these pill bugs. We're going to look at their phototaxis and their hydrotaxis. So I'm going to put those in the chat. What do you guys think those might have to do with? If we know some parts of what these words mean, what do you think? What do we think hydro or photo might refer to? Oh yeah, water. Hydro, right, means water. So hydrotaxis refers to whether a pill bug will move toward or away from water, exactly. For photo, exactly. Photo means light. So a phototaxis is, does the pill bug move toward or away from light? So we're going to kind of create a testing chamber to see how some of our pill bugs react. And I should say before we start, I chose pill bugs just because they're kind of easy to find. A lot of us have them in our yards, and they're easy to pick up, right? And they don't transmit any kinds of diseases or anything like that. So they're a great subject to study with. They're also fairly resilient, but this is something that you could study with any kind of insect, right? As long as it's safe for the insect and it's safe for us. Insect, bug, any other kind of small organism, right? So we're going to talk about specifically pill bug taxis by creating this testing chamber. Before we start, can I ask, are any of you guys going to be doing this experiment along with us at home? I'm always interested to see. So some of us are. Oh, that's cool that some of you are. And if you're not, that's totally fine. Sometimes it's fun to watch someone do it first, and then you can try it on your own time. Yeah, and sometimes it can be hard to kind of do everything in the short amount of time that we have. Totally understand that. But I am excited to do this experiment with some of you, and excited to show the experiment to the rest in the hopes that maybe you will check it out later. So I'm going to go ahead and switch my camera to the experiment cam. And I'm going to show you what I have here. Here is my experiment cam showing this plastic bowl, right? And in this plastic bowl, what do we see? Maybe I can even bring it a little bit closer. Because what I see are some dead leaves, some dirt, leaves, yeah, exactly, you guys. And then I see a wet paper towel, yeah, exactly, that I put in there. I'm gonna go ahead and take the paper towel out, whoops, and on this paper towel is one of our test participants here. I'm going to put it back into the bowl really gently. And actually, it's doing a pretty cool behavior that you guys may have seen before. What is it that roly-polies do when they get, it's kind of the reason we call them pill bugs or roly-polies. What do they do if they feel threatened? Yeah, they roll themselves into a ball. They roll up, exactly. Oh, was there another one on here? Oh, there was, good spotting. So, right, that's another, maybe a very different kind of taxis that they perform. I'm not sure if we would refer to it as that, but it is definitely a behavior that they do. That's a great point there. I'm going to take out some of these leaves just so that we can see our pill bugs a little bit better here. And some of them are on their backs, but they'll be able to flip themselves over pretty easily. So what I'm doing is I'm clearing my test area here because you want to make sure that you have kind of the beginnings of your chamber. If we want to test different conditions, different environments, right, then we want to make sure that we have a place to do that testing in. So I have my bowl here. I have my test subjects. I'm not sure that I counted them. I told you guys to find maybe at least 5, just so that we could get an idea of what multiple individuals will do, right? Because we can test just one, but it's better to learn what more than just one organism will do, right? So I've got a couple here, along with a few little pieces of leaf litter. But I've got my bowl, And I've also got a little piece of paper. And this is something that we don't have to use, right? But it's something that might help divide our testing chamber in half. So I have a piece of paper, and I measured it to be about the size of my bowl. And then oops, we have a little traveler on here. I included kind of this little cutout so that there is a door. I want to make sure that there are 2 of these testing chambers, so I'm splitting it in half with my paper. To keep it in place, I have a little bit of tape. So you can do this part if you would like. I'm just going to tape this paper in place. Now I've got some of my roly-polies on one end and some on the other, and that's okay because they'll decide where they want to go, or they will perform that behavior, that taxis, to decide where they want to go. So now what I have is 2 different paper towels. And right now I'm saying that they are 2 dry pieces of paper towel. And these are pretty small, right? If I'm testing hydrotaxis, what do I want to do to one of these pieces of paper towel? What do you think? What should I do? If I want to test whether they go toward or away from water, exactly, I could get one of these wet. Yes, so I'm going to take a piece of my other wet paper towel here. So I've got a piece of paper towel that is wet, and I've got one that is dry, right? So I'm going to put one on each half. This is going to be my wet side, and this is going to be my dry side. And I noticed that I have a bunch of roly-polies over here on the dry side just because that's where they wanted to go, but that's okay. I will let them decide where they want to go. So right now, their environment has changed. And one way that I could really kind of keep the environment a little bit smaller so that they really register that change quickly is to put a piece of plastic wrap over the top of my bowl. This plastic wrap makes it a little bit hard for the camera to see what's going on inside of it, so I'm not going to put it on there. So they might react slightly more slowly, but we're going to see what they do. I want you guys to do something with me now. This is my wet side, and this is my dry side. I want you guys to make a guess. Do you know what it's called when we make a guess in a scientific context? What do we call a science guess? This is one of those science terms that you'll see a whole lot. What do you think? Any ideas? Ooh, yes, a hypothesis. So we're making a prediction or a hypothesis about whether the pill bugs will go toward the wet paper towel or the dry paper towel. Do you guys have any guesses? Do you have any predictions? You can just say wet or dry. What do we think? Do we think they'll want it to be wetter, drier? And this isn't about being right or wrong. Making this guess is just to kind of give us an idea what we're looking for and to get our brains started thinking scientifically. So this is really great. I'm seeing a lot of different guesses. And we are going to find out what they do by watching them. Now, again, they're going to kind of wander around a little bit to get used to their surroundings, and we'll see if over time they do prefer one side or the other. I'm not really sure which one they will do. There's a lot of reasons why they might prefer the wet over the dry or the dry over the wet. We'll just have to see, because that's how we figure out what a hypothesis is. Now, what I'm going to do is while we are watching them move around a little bit, I am going to use this special camera that I have called a dino light. And what it has is a bunch of lights in there, and it also has a tiny camera. And this cool plastic part here kind of creates a field of vision, so it makes it so that if I put it down on a surface, it doesn't, it doesn't kind of crush anything between the camera and the surface. So I thought it would be really good to use to look at some of these roly-polies up close. You guys want to check them out up close? They're really, really interesting looking. While they're moving around, I'm going to try not to bother them too much. But I'm going to go to my dino light cam, and it's going to be blurry, but I'm going to focus it in. All right, so right now it's a little bit blurry, but I'm going to focus it in on some of my roly-polies. Do you guys see them? Whoa. So can you tell me some things that you notice about them? I'm gonna try and zoom in a little bit here. Ooh, there we go. Oh, here's one. Can you tell me something that you notice about these guys? What do you think? Oops, there we go. Yeah, they're kind of spotted. Now, some of them have like a little bit of dirt on them, but I noticed that some of them have different patterns. And I would have never known that if I hadn't looked more closely. Let's see if I can get this guy. Ooh, there we go. Check that out. They have a little bit of a pattern up close. There was one in here that actually had a little bit of almost like a green pattern. Maybe it's this one right here. Yeah, look at that. I have never seen that before. So when I was looking at them earlier, I was just amazed. They do appear, that's a great observation. They appear to have something kind of like scales, right? They have this really cool armor that helps them, that helps protect them against stimulus in their environment that they would want to escape, right? Like a predator or something like that. Someone else noticed that they have lots of legs. They certainly do have lots of legs, that their patterns are almost stripy. I have one in here. This one is kind of light colored, and I'm not really sure why. I don't know if maybe it's a younger one, so it has a lighter color. Then I have ones that are really dark, and those do happen to be my bigger ones. So I wonder, I would have to look that one up. I'm not totally sure. So let's check here. We've got a bit of movement. They're exploring the piece of paper, right? We've got a couple of them hanging out on the dry paper towel, checking it out. And then on the other side, the wet side, we've got some crawling around. We've got some wandering. They're just kind of checking it out, right? I'm gonna go ahead and move my camera back for kind of the last part of this experiment. Yes, you can see the antenna up close. That's a great observation. They use those antennae to feel around in their environment. We're going to test one more thing with our roly-polies. We're going to see, we're going to continue letting them see if they prefer the wet or dry environment. And we're also going to create, we're going to test that phototaxis. So we're going to create half of the environment that is dark and half that is light. So to do that, I just have this towel, and I'm gonna turn us back to the experiment cam. Oh, this is a great observation. They look kind of like mini armadillos. That's true. They really do have some of those similar overlapping scales. So I'm gonna gently push some of these guys back into the testing chamber. This is when the plastic wrap over the top would be helpful, but that's okay. What I'm gonna do is I'm gonna take my towel and I'm gonna cover up half. Now I don't wanna cover up just one half here or one half here, because I don't want to interfere with the test about water. I don't want them to actually not like the dark and only like the dry, but they still come over here because it's dry, right? I just wanna test one thing at a time. So I'm gonna kind of lay it down, we would say perpendicular, meaning crossed like this. Our chamber splitter is going around this way, and we're going to split it up again this way. So I have created a dark half here and a light half here. But they're also still dark and wet chambers, so it's kind of testing both phototaxis and hydrotaxis at the same time. I'm going to ask you guys to make one more hypothesis with us. Yeah, do we think that they will go to light or dark? And this is an even bigger hypothesis. Do you think that they will go to light and wet, dark and wet, light and dry, or dark and dry? What do we think? We have a lot of different ideas, yeah. There's so many different kind of combinations, right? So I'm gonna give them some time to perform their taxis, to go wherever they're going to go. I'm seeing a reaction to light a little bit faster, I think, than I'm seeing to wet and dry. And that might be because their environment is open, so that wetness and dryness is not as contained. Oh, this is really great. I like this. So we have a viewer who shared, Cecilia, who shared that perhaps they will like it dark and dry because they will like to hide under rocks. I definitely did find these guys under some rocks. So maybe that is something that they will react to. Does anybody remember when we were talking about them at the very beginning, what animals are they most closely related to? What other animals? I'm going to go back to the dino-like can. Yeah, so they're really closely related to crabs. They're what we call isopods. Oops, there's one right there. So if we know that information, does that change what we might guess about whether they like wet or dry? What do you think? It might inform our answer to say, like, well, so maybe if they like, maybe if they have gills like crabs and they need moisture in their environment, maybe they would prefer it to be wet. Although it's true what you guys said, that they don't like to be submerged in water. They don't like to be entirely put into the water. They cannot breathe like that. They just need wet, moist air. So I'm going to go ahead and switch back to my experiment cam just in the moment we have left. Experiment cam. And I'll let you guys take a look. They're still kind of moving around, but we've got some in our light side. We've got some on the towel here. I'm going to push it off there a little bit. And I'm going to uncover it. And what do we think? Do we see more on this side or this side, where it was light or where it was dark? It's a little hard to tell now that I've taken it off. What do you think? I don't know. I think it's a little bit hard to tell, but maybe they did slightly prefer the dark. Maybe slightly. I think that probably this experiment takes a little bit more time than I've had here today to let them settle into their new environment and decide where they want to go and properly respond to their different environmental changes or stimulus. But we have at least set up our experiment that you guys can always check out yourselves. And we've also been able to take a look at them up close. Take one more quick look at them up close. That is so cool there. Sometimes they're just hanging out. Sometimes they're pretty happy with their environment, so they stay put. This one flipped over. So our experiment, I would say, is not finished. I think that they need a little bit more time, but I'm really interested to see what your roly-polies might decide, because some roly-polies prefer the dark, right, because they usually live underneath the leaves and rocks, but some of them also have a certain condition. It's actually a type of virus, although humans cannot catch this virus. It's only for roly-polies. That makes them move toward the light. It just changes their behavior to make them move to the light instead of the dark. So I don't know if maybe some of these guys have that condition where they would want to move closer to the light rather than the dark. I also don't know if maybe they're looking for something to eat and maybe they think about a dry leaf being more like their food rather than something wet. So possibly they were going towards that dry paper towel, having that in mind. I don't know. It's something that I would have to experiment more with them to see what happens. Oh, this is another great comment that the sun is shining more on one side, so maybe that is changing the outcome. That's a great point. Definitely using like a lamp or some other light source might help us as well if it were coming from the top rather than slanting from the sides. That's a really great idea. You guys are so full of these amazing ideas. You all had so many great hypotheses, and even if we were not able to prove them today, you all can do this experiment at home and see what your results may be. It's always better to have more data, so I would love to know what you guys find. That is all the time that we have for today. So I want to thank all of you for being here. If you want to share your experiment or any other ideas, you can always post it on social media and tag the museum and hashtag Lab at Home. We would love to see what kind of stuff you guys come out with. Next week, we will continue our summer bug out celebration with experiments inspired by bugs that live in water. So if you're interested in bugs that live in water, we've got some experiments next week that are going to be pretty cool. As always, we will send the curriculum for this week's Lab at Home via email by the end of the day today, and you can also watch this recording of our live stream on the museum's on the museum's YouTube channel later on in the week. I want to thank you all so much for being here today, and we hope that you will join us again next week. Labs are every Wednesday at 2 p.m., and you can register on our Life and Science at Home page. So I want to, again, thank you all for being here. Good luck finding more roly-polies, or pill bugs, potato bugs, wood louses, whatever you decide to call them. And I wish you luck also in your experiments. Thank you all so much for being here today. I'm gonna let this little bug say goodbye to you as well. See you later, guys. Thanks so much. See you next week.